Comparative analysis of cognitive function of CL 2006 and wild-type C. elegans strains. Alzheimer's disease is a chronic neurodegenerative disorder that is the #1 cause of senile dementia. Out of 5.2 million Americans that have Alzheimer's disease, 5 million of them are over 65 years old. Over $293 billion have been spent because of Alzheimer's disease in 2013 alone. Over 83,000 people have died from Alzheimer's disease in 2010. That is like 27 people dying out of every 100,000 people. During the last century, there has been a dramatic increase in the life expectancy of the American people, from nearly 47 years to more than 78 years. Given that age is one of the most significant risk factors for developing neurodegenerative diseases, more individuals achieve an age at which neurodegenerative disorders are common. Now, since there is such a spike in the life expectancy age and since Alzheimer's disease is more common in people that are older, it is inevitable that the cost of taking care of people with Alzheimer's disease will dramatically increase. Therefore, it makes sense why research in Alzheimer's disease has exploded, all in an effort to discover new ways to treat and prevent this disease. It is felt that Alzheimer's disease occurs when clumps of proteins or amyloid plaques or tau tangles grow in the brain. The protein strands twist and damage brain cells, and the brain cells die and certain areas of the brain shrink. This slide represents the pathological findings found in the patient with Alzheimer's disease. These include a shrunken brain and large ventricles, dying neurons with tangles, and plaques made of beta-amyloid peptide. Research has focused on many different venues in an attempt to try to treat and control Alzheimer's disease. The challenge has become to try and prevent the disease and its cognitive decline. In the second half of the 20th century, C. elegans was introduced as a model to study development and neurobiology. Today, C. elegans is used to study a much larger variety of biological processes. Many key discoveries, both in the basic biology and medically relevant areas, were first made in the C. elegans model. This slide shows the life cycle of the C. elegans, and make note that it is the L4 stage that is most useful to most studies on C. elegans. Researchers were able to create a transgenic strain, the CL 2006, which possesses the AB peptide, which is found in Alzheimer's disease. Studies suggest that this new transgenic strain is a suitable in vivo model to relate AB expression with its toxicity, which may correlate with Alzheimer's disease pathology. It has been used as a pharmacological evaluator for novel therapeutic agents. These studies have looked at the effect of these drugs on the AB peptide expression and toxicity. However, we must remember Alzheimer's disease alters the cognitive function of the individual. Our question then is, does the transgenic CL 2006 strain that possesses Alzheimer's B-amyloid peptide also possess altered cognitive function? So, we have set out to see if there is any measurable change in the cognitive function of the transgenic CL 2006 strain than that of the wild-type strain. Therefore, the hypothesis is, if the CL 2006 strain possesses Alzheimer's B-amyloid peptide, then it should also express altered cognitive function. Prior studies have shown that cognitive function can indeed be measured in the C. elegans model. Nematodes including C. elegans respond to touch, taste, smell, and temperature, and other Model Ts all project into a single circuit that is so-called the head navigation circuit or the nerve ring. How this circuit integrates these inputs to generate appropriate behaviors is considered to be the nematode's cognitive function. The most well-established protocol to measure cognitive function is the olfactory sense, smell. Multiple studies have used this sense to document the normal responses of the C. elegans to both attractants and irritants. These next two slides summarize the list of attractants and repellents that have been used to map the cognitive responses of the C. elegans to their exposure. These two graphs show how the C. elegans reacted to different attractants and repellents as measured by a chemotaxis index. This shows the simple protocol used in order to determine the chemotaxis index, which is used to quantify cognitive function from the C. elegans to attractants and irritants. This slide shows that an attractant sodium chloride is placed at one end of the Petri dish. The C. elegans in the L4 stage are placed in the middle, and in a fixed period of time, the chemotaxis index is calculated. The chemotaxis index equals the number of worms of the attractant minus the number of worms of the repellent divided by the total number of worms in the assay. Since we are using the chemotaxis assay and the smell on a stick assay, which is based on the ability of the worms to move, keeping in mind that the transgenic CL2006 strain becomes paralyzed at high temperatures, the two assays, chemotaxis assay and the smell on a stick assay, need to be performed at 16 degrees Celsius. This will prevent the known paralysis that occurs in the transgenic strain at 25 degrees Celsius that would affect the chemotaxis index. This is the flowchart that summarizes the experiment. The organism that will be used for this experiment is the C. elegans, which will be grown in growth agar plates with OP50S food. We will perform the chemotaxis assay and the smell on a stick assay for both the wild-type strain and the CL2006 transgenic strain at a temperature of 16 degrees Celsius. The analysis that will be used is that there will be a statistical analysis run on SPSS along with an ANOVA post hoc Scheffé, and P will be less than 0.05. The chemotaxis assay. Animals are placed on a large petri dish on which a spot of volatile attractant and a dilutant have been placed on opposite sides of the plate. A drop of sodium iodide, which paralyzes the animal, is also placed with the odorant and dilutant. Animals are placed in the middle of the plate and are allowed to crawl around freely for about 1 hour. The smell on a stick assay. A paintbrush hair is dipped in a volatile chemical repellent and presented to the anterior tip of a forward-moving C. elegans, where the amphid pore is located. The hair is not allowed to touch the animal. C. elegans typically respond rapidly by reversing direction, usually within seconds. When creating the plates for the C. elegans, 10 milliliters of liquid NGM should be placed into each petri dish and be left to sit overnight. Then, the next day, when preparing the C. elegans plates, add 250 microliters of E. coli strain OP50 into each plate as food for the C. elegans. This is basically all you need in order to keep the C. elegans alive. In summary, we look to show that the transgenic CL2006 strain not only possesses the physical findings of Alzheimer's disease, the B-amylase peptide, but also altered cognitive function when compared to the wild-type C. elegans.